So this video is going to go through one of the level 4s on post-transcription modification. So this is going to focus on what happens directly after the RNA is transcribed. So let's take a look at the beginning picture here. So we're looking at DNA, so I've got my A's, T's, G's, and C's. When I look at the original gene here, I notice that it's made up of these exons and introns, and they kind of alternate back and forth. My promoter region is basically where we're going to start creating my RNA sequence. When I first do transcription, I'm going to end up with my primary transcript. So my primary transcript is kind of my rough draft. It's my first version of my RNA, and we're going to do some modifications to it. So when I look at my primary transcript and what happens next, you're going to notice that my introns, which are these red sections, these get removed, and my exons, which are these blue sections, those are what are actually going to remain. So what ends up happening is my introns are sections of DNA that are not going to be read. So my introns are actually cut out. However, my exons are the portions that are going to be expressed. So that's why they're called exons. Not because they're exiting, but because they're being expressed. Those are the sections of DNA sequences that we want to use when we code for the protein. So this process is called RNA splicing. So they call it splicing because basically you're kind of slicing, I'm slicing the RNA into these little sections. And then I'm cutting out my introns, so those guys get removed. And then I put back all my exons back together again. So RNA splicing is one type of post-transcription modification that happens. There are two more things that are happening to kind of help protect the mRNA before it leaves the nucleus. So on one end, you're going to place something that's called a methylguanine cap. Guanine cap. And that's meant to kind of protect that. And at the other end, we're going to add something called a poly-A tail. And what we're doing is these are just tons and tons and tons of adenine bases or adenine nucleotides. And the purpose behind this poly-A tail is that once the mRNA leaves the nucleus, it's going to be attacked by enzymes that are eventually going to break it down. So by having a longer tail, it actually increases kind of the lifespan or the likelihood that it's going to remain unchopped before it ends up getting attacked by enzymes. So once we've done these two things, we've spliced the molecule and we remain with exons only, and we put this cap at the end, and we put the tail, poly-A tail, at the other end. Now your mRNA is complete, and it can leave the nucleus and enter into the cytoplasm, where this mRNA is going to be used for translation. We're going to use this model to actually make the protein.